Hello, and welcome to this new Genetic Engineering and Biotechnology News webinar entitled Biology by Design, Applying Gigabases of DNA to Bioengineering. I'm Jeff Bogaliskis, Technical Editor for GEN, and I'll be the moderator for today's presentation. On-demand cell factories, cannabis from yeast, or entire living cells created from scratch. These are just a few examples of the innovative ways synthetic biology is gearing up to shape biotechnology. Yet, engineering these biological systems used to be a laborious and expensive undertaking, especially when the end goal required large amounts of DNA synthesis. Now, DNA synthesis can be achieved for pennies per base pair, and investigators are no longer required to undertake traditional cloning methods, allowing for novel bioengineering techniques. In today's webinar, our speaker will tell us why DNA synthesis is fundamental to the efforts of Ginkgo Bioworks, the Boston-based company focused on organism design, and how their partnership with Twist Bioscience has allowed them to design an array of engineered microorganisms. So let's meet our guest presenter for today's event and get to the exciting data he has to show us. Patrick Boyle is the head of Codebase at Ginkgo Bioworks. Patrick is responsible for the company's portfolio of reusable biological assets known as Codebase. Assets that include novel strains, enzymes, genetic parts, and diverse genetic repositories. In today's presentation, Patrick will discuss some innovative synthetic biology applications for bioengineering, as well as Ginkgo Bioworks' collaboration with Twist Bioscience, who has agreed to supply the company with more than 1 billion base pairs of DNA.
Hello, everyone. My name is Patrick Boyle, and I'm the head of Codebase at Ginkgo Bioworks. In today's presentation, we'll be talking about our large-scale DNA synthesis work done in collaboration with Twist Biosciences, and specifically, I'll be talking about how we leverage DNA synthesis to accelerate the bioengineering process. I'll kick things off with a brief intro to Ginkgo, including our philosophy for bioengineering, as well as a preview of some of the projects we're working on in our foundries. Next, I'll discuss why you should consider DNA synthesis versus other means of building DNA. I'll also discuss how Ginkgo and Twist use software to make this process manageable at large scale. I'll then go over some case studies that illustrate how we put large-scale DNA synthesis into practice to accelerate the bioengineering process. Finally, I'm looking forward to answering your questions about the application of DNA synthesis to your research challenges. Ginkgo is based in Boston, and our origin extends back to the founding of synthetic biology at MIT. This is Tom Knight, who's one of our founders at Ginkgo. He spent his first 30 years at MIT as an electrical engineer. In the mid-90s, Tom realized that in the same way that computers had revolutionized the 20th century, biology had the potential to revolutionize the 21st century. Tom realized that at its core, biology is digital code. So information in the form of DNA can be translated into a nearly endless array of products. The challenge, of course, comes in learning how to write that code. At Ginkgo, our mission is to make biology easier to engineer, and we're doing that by developing engineering principles for biology over the course of dozens of bioengineering projects. Here's a snapshot of one of our foundries. In this photo, you can see that we heavily rely on automation to do our work. By removing many of the manual steps that are typically used to do wet lab work, we can better design and analyze our experiments via software. In many ways, our foundries are rapid prototyping facilities, supporting tens of thousands of designs per month across many different projects simultaneously. At Ginkgo, our foundries have specialized teams working on the different aspects of engineering biology. In effect, the foundry brings under one roof all the tools needed to design, build, and test engineered organisms. At Ginkgo, we support many different microbes as well as mammalian cells in our latest foundry. That means that these processes have to be flexible to support a wide variety of projects. Large-scale DNA synthesis is transformative in this type of environment because it effectively debottlenecks the build part of the design-build-test equation. That means that we can focus on scaling our design and test tools. And while I don't have the time to talk about this today, we also extend our work to include highly automated fermentation, which helps us graduate the best strains to manufacturing as quickly as possible. Most projects at Ginkgo fall into three main categories. For chemicals projects, we're engineering microbes to produce specific small molecules that our customers have requested. For strain improvement projects, we're working with our customers to take existing fermentation strains used in industrial applications and improving their performance. And then finally, we also have projects to engineer organisms to produce enzymes. By focusing on automating the bioengineering process, we've been able to leverage our foundries to help customers across many different markets. So for example, in partnership with Bayer Crop Sciences, we've launched Join Bio. Join Bio is a joint venture that engineers microbes to fix nitrogen for crop plants, replacing traditional fertilizer. By automating much of the microbial engineering work at Ginkgo, that frees up the Join team to focus on testing those microbes on plants and getting their products out to market faster. In the pharmaceutical space, we're partnered with Synlogic, a company that engineers E. coli to serve as living medicines in the gut. We work with Synlogic to rapidly prototype and improve their microbes in much the same way that we engineer microbes for Join in the agricultural space. We also have a partnership with the Kronos Group in Canada to engineer microbes to produce cannabinoids. In addition to the well-known molecules THC and CBD, there are many other cannabinoid molecules with the potential to be valuable in a range of pharmaceutical applications. Unfortunately, many of these molecules are only present in trace quantities in the cannabis plant itself. So what we're doing with Kronos is engineering microbes to produce these cannabinoid products via fermentation. In the food space, we just recently announced Motif, a new spin-out company that's focused on the production of food ingredients, particularly alternative proteins. In all of these collaborations, Ginkgo is focused on delivering novel microbes to our customers as quickly and reliably as possible. It's therefore critical to us to integrate technologies that are scalable, dependable, and fast. Okay, so that was an extremely quick tour of the type of projects that we're working on at Ginkgo. I'd like to switch gears now and talk about why we think gene synthesis is so important to our growth, and also to the growth of the broader biotech industry. Given the broad applications we've found for DNA synthesis at Ginkgo, it's likely that you have ongoing work in your own labs that could be improved via gene synthesis. You may be familiar with Moore's law for semiconductors, which set the tempo for improvements in the electronics industry for more than 50 years. Over the past 15 years, we've seen similar improvements in our ability to read and write DNA. Probably everyone listening today is familiar with the dramatic cost reductions in DNA sequencing over the past 15 years, as shown on the graph on the right. Sequencing a human genome, for example, now costs hundreds or thousands of dollars rather than hundreds of millions of dollars as it did in the year 2000. That inflection point that you see in the sequencing curve is the advent of Illumina technology. You may be less familiar with the fact that DNA synthesis has also been rapidly improving in cost over that same time period. In fact, sequencing and synthesis have improved at a rate faster than Moore's law over the last 15 years. As a synthetic biologist, this represents an exponential improvement in our fundamental ability to program DNA. So both reading and writing code is becoming cheaper every year, meaning that we can do more synthesis and sequencing every year. At Ginkgo, we scale the output of our foundries based on the fact that we can read and write more DNA each year. So this is directly analogous to what happened in electronics. Software developers knew that chips would get better every year, so they pursued more and more ambitious software projects based on Intel's roadmap. The graph on the left shows how the output of our foundries has improved over the years, again with exponential growth driven by, among other things, improvements in our reading and writing of DNA. How do we think about incorporating DNA synthesis into our work at Ginkgo? In short, we're looking forward to the day when hand cloning of DNA goes completely extinct. We have multiple in-house services to deliver DNA to our organism engineers. If at all possible, DNA at Ginkgo goes straight from twist, delivered as sequence-perfect clonal DNA, to transformation into a host organism, and onto testing from there. Other services make use of non-clonal linear DNA fragments from twist, which aren't sequence-verified but can be quickly assembled in an automated fashion for further use. Here, we're trading the convenience of getting sequence-verified clonal DNA for the flexibility and speed of getting non-clonal fragments. Even in this case, our preference is to leverage automation to reduce the amount of low-throughput bench work that's needed to make DNA. For groups that do large volumes of cloning today, or have experiments that could benefit from more DNA diversity, it makes a lot of sense to systematically outsource that cloning to synthesis, particularly when you factor in the cost and effort to sequence-verify clone constructs. For small volume cloning, it still makes a lot of sense to free up FTEs that could be working on other experiments. Personally speaking, I think I spent more than half my PhD doing by-hand cloning, and I imagine the molecular biology community as a whole loses lots of productivity to cloning. Nobody does by-hand Sanger sequencing anymore, and I think cloning will be similarly obsolete soon. So how do we actually order DNA at Ginkgo? Of course, Twist has a website with a graphical interface for ordering DNA that most users will use. In our case, however, we routinely order more than 10,000 constructs from Twist every month. So there's simply no way that we could do that via a conventional user interface. To keep up with these order volumes, Twist developed an API that allows us to order DNA programmatically. This is integrated with our software here at Ginkgo, but Twist has also opened up the API for use by other customers. Here's a cartoon schematic of how large-scale ordering of DNA from Twist works here at Ginkgo. I'll walk through the process at a high level in the next few slides, but if you're interested in the details of how the Twist API works, I've also included a link to the API spec below. The ordering process starts with the design on the Ginkgo side. So we have custom software that our organism engineers and designers use to choose a library spec and to generate the appropriate DNA sequences. These sequences get stored in our sequence database that's part of our laboratory information management system. For a sense of scale, we've chosen to receive all DNA from Twist in the form of barcoded 384 well plates. So a small design by our standards would be around about 100 designs. We then send the sequences from our limbs over to Twist via the API. A number of things happen before an order is confirmed. First, the API runs a check to make sure that the requested DNA can be synthesized, and whether or not the requested cloning strategy is supported by the vectors that we've onboarded at Twist. Once these checks are passed, the order can then be quoted and confirmed. Following this process, we regularly pull the API to get updates on manufacturing status as well as shipping dates. As DNA ships to us, we've set up software on our end to log plates as they arrive. Our DNA receiving team can pull all the necessary information such as plate maps and barcodes directly from the API. We also use the messaging service Slack pretty extensively at Ginkgo, so our software team wrote a notification service that sends a Slack message directly to people as soon as their DNA has arrived. This process has turned out to be very robust, so we've battle tested it on tens of thousands of sequences so far. You simply can't order DNA at these volumes via traditional websites. Even with an Excel sheet uploader, which is probably the gold standard currently for high-throughput DNA ordering, that just wouldn't work here. So for one thing, the file sizes would be too big. And at our volume, ordering sequences via manual entry or even automated entry into an Excel sheet would be way too error-prone. So with an API, instead, we're given a discrete set of endpoints that our software can interface with. And having those discrete endpoints means that Twist can make big changes to their software without negatively impacting our use of the API for ordering. Likewise, the Twist API doesn't care what algorithms or software that we're using on the Ginkgo side to generate DNA sequences, so we're free to update our methods as often as we need to, and that doesn't impact ordering. If your group or company is looking to order hundreds or thousands of constructs from Twist, I highly recommend checking out the API. Particularly for computational biologists who are used to working with APIs, it's a pretty straightforward way to order DNA from Twist. We like the flexibility that the API offers. So for example, we can bypass the Twist codon optimization process in cases where we've already applied our own optimization algorithm. Multiple teams can also use the Twist API to get the information they need asynchronously. So for example, the folks on the build team can check in to get plate maps and other data without needing to check in with the person who originally ordered the DNA. And since all of our DNA arrives in 384 well format, it saves a lot of time to get all the plate data systematically and associated with barcodes. With the number of projects we have going on, the API is really handy for keeping up to date with individual orders, and it allows us to set up notifications so that people know right away when their DNA has arrived. Now I'd like to talk about some of the ways that we use large-scale DNA synthesis at Ginkgo. Given the variety of projects and organisms that we support, we've been really happy with how often we're able to use DNA synthesis to get better results. As a reminder, all of this work is happening in our highly automated Foundry facilities. However, even if you're using very little automation to do your work, by hand cloning is still likely to be one of the rate-limiting steps in your process. Here, upstream of DNA synthesis, we put a lot of development effort into developing bioinformatics and design software. Downstream of synthesis, we've invested in a wide array of methods for high-throughput screening. In both of these cases, ordering more DNA makes it more likely that we get successful results. In other words, as DNA gets cheaper, it will make more and more sense to take advantage of all the available screening capacity with more designs. Twist has a number of different product offerings, but the easiest drop-in replacement for cloning by hand is to order sequence-verified constructs and plasmids. When you're used to cloning DNA by yourself, waiting 15 days for constructs to arrive can seem like a long time. In our experience, however, the fact that batch sizes for synthesis are effectively unconstrained means that you can save time overall by increasing the number of designs that you test in each round. Traditionally, you may iterate on a small set of designs successively until you find the design that functions as you expect. On the other hand, if you take the time to generate a larger number of designs that test a range of hypotheses in one experiment, we found that we save a lot of time overall. This doesn't even factor in the productivity savings you get by replacing time spent cloning or sequencing with other work. In these case studies, we've used design tools developed at Ginkgo to generate all of the DNA. Because we rely on large-scale DNA synthesis, our design tools are also focused on large-scale designs. There are a number of benefits to us for doing this. So we can collect data sufficient for training machine learning models that we use to make our design tools even better. We also use flexible compute infrastructure, including Amazon Web Services, to do more high-throughput computational design than we would otherwise bother to do if we were designing small numbers of constructs. And as with the API, much of the work that we're doing is too high throughput to do with traditional graphical DNA editors. So we've written software and scripts to automate our genetic design. Tools like Jupyter Notebooks make it easier for computational biologists to rapidly prototype and share design strategies. All of this infrastructure depends on the fact that we can generate thousands of genetic designs via synthesis. If we were doing smaller scale experiments, we wouldn't realize the benefits of these design tools at all. Here's an example of how we've used large-scale synthesis for enzyme discovery. In this case, the enzyme class we were working with was known to produce a mixture of two products. Multiple groups had tried to engineer the canonical example of this enzyme to make it more specific, but were unable to completely eliminate the undesired activity. The map on the left illustrates a set of more than 5,000 potential enzymes that we identified as candidates for this enzyme activity. As sequencing has gotten cheaper, sequence databases have been flooded with thousands of poorly characterized or outright misannotated enzymes. This map was generated by our design software and helps us decide which enzymes to synthesize in the hopes of finding enzymes with better performance. This particular enzyme played a key role in one of the pathways we were engineering. So the fact that the enzyme produced a mixture of products meant that we were losing some carbon flux that ideally would be converted to our final product. This map clusters together enzymes based on predicted function. In this case, the canonical version of this enzyme, the version that produced two products, is a member of the cluster shown in green. Two other clusters, shown in blue and red, included some enzymes that had been shown to have similar enzymatic function but on different substrates. Some of the uncharacterized enzymes in these clusters may in fact have activity on our product of interest. In orange and purple, we have clusters in which we had no known enzymes to compare to. So these clusters contain primarily enzymes deposited in sequence databases from metagenomic sequencing surveys with essentially no activity data available for them. I've now overlaid this map with black dots, which represent the enzyme sequences that we selected for synthesis. In this case, that's well over 1,000 unique sequences. To give you a sense of where these enzyme sequences are coming from, the map on the right shows the same enzymes, except they're now color-coded based on the type of species that they came from. As you can see, this library is extremely diverse. Because we're using DNA synthesis, we can cast a wide net to see what sequences may encode the enzyme activity that we're looking for. This approach is more ambitious and also less biased than any cloning-based approach to enzyme discovery because we're not dependent on the availability of physical DNA to clone from. We just need the digital sequence. Furthermore, we can take the time to re-encode the nucleotide sequences of all these enzymes so that they are more likely to express in our screening host. Because some of these clusters contain enzymes that have been characterized to have the activity we're looking for, or at least a similar activity, we decided to most heavily sample those clusters via synthesis. However, since the known enzymes have been shown to be nonspecific, we also lightly sampled the more distant clusters in the hopes that they may reveal enzymes with the correct activity and ideally improved specificity. We're often surprised by how poor automatic annotation can be for these types of metagenomic sequencing datasets. So for example, in some of our previous screens, we found enzyme hits that were originally annotated in databases as transcription factors. Obviously, not all of the enzymes that we'll test in these experiments will have the activity that we're looking for. That being said, even negative data is extremely informative for our data scientists. So, for example, clusters of enzymes that don't show the activity that we're looking for can be excluded from future experiments. Similarly, residue level differences in protein sequences that lead to decreases in activity can be avoided in follow-on protein engineering campaigns. We can also recombine features of the most active enzymes to further improve their activity. One of the many tools that we use at Ginkgo that I won't have time to discuss in depth today is metabolomics. We put a lot of effort into developing high-throughput mass spec methods so that we can collect metabolomics data for many of our enzyme screens. This was very valuable in this particular case, where the canonical version of the enzyme that we were working on made two products. Via metabolomics, we were able to measure the production of each of these products across the full library of over 1,000 unique enzymes tested. This slide shows that same map of enzyme diversity that we sampled via synthesis, now with the actual experimental data overlaid on it. The map on the left includes a heat map showing which enzymes showed the desired activity, that is, the enzymes that produce the product that we were looking for. The map on the right includes a heat map showing which enzymes were more specific for producing that product of interest. I've now highlighted the cluster that contained the original sequence that we were working with. That cluster contains many variants that also have that activity, which is to be expected because we've clustered them together based on sequence and structural similarity. As you can see on the right, many of the enzymes in this cluster appear to be highly specific for producing just a product of interest as well. In another cluster, we see enrichment of sequences with very high specificity. This is exciting because none of the previously characterized enzymes in this cluster had the activity that we were looking for when we started. These sequences are also quite distinct from those highlighted in the previous slide. Finally, even the most distant clusters in our model yielded active enzymes. These candidates could be very interesting for further protein engineering given their distance from the other characterized enzymes. Here's a plot showing activity versus specificity for the highlighted enzymes on our map. The enzyme that we started with produced the desired product with 80% specificity. That is, it produced an 80-20 mix of the two products that we were measuring. As you can see, we identified novel enzymes with 100% specificity for a product of interest after this screen. Many of these enzymes are also nearly as active as the enzyme that we started with. Some of our best enzymes in this screen have as little as 20% sequence identity to our starting enzyme. Even extreme protein engineering campaigns are unlikely to have sufficient sequence diversity to find enzymes like that. In addition, engineering enzymes for specificity is very challenging. In this case, we identified enzymes that lack the off-target activity altogether. This screen shows how useful synthesis can be to allow us to quickly identify better enzymes and other genetic parts. All of this screening was done following a single round of synthesis at Twist. Screens of this scale are now quite routine at Ginkgo. This slide shows another screen for a different enzyme activity. In this case, I've plotted the enzyme library via a dendrogram, which can help illustrate highly diverse libraries. For example, you can see two boxes in this figure. The box in the lower left represents enzymes from bacterial species. The box in the upper right represents enzymes sourced from eukaryotes. The columns to the right of the main figure show the similarities in domain structure among the enzymes in the library. Further to the right, we can see a column that represents which enzymes were synthesized. In this case, we synthesized the enzymes in 3 different batches. The following columns plot our work to test these enzymes in multiple rounds of screening, narrowing down to the best candidates. In this case, we needed to modify the N-terminal sequence of these enzymes for secretion. So we synthesized 2 different versions of each enzyme, representing 2 different N-terminal design strategies. This slide shows the type of dramatic improvements that we've achieved via this approach. So the arrow in this case is pointing to the enzyme that had previously been characterized as the best-in-class variant of the enzyme, and this is the data from the library that I showed in the previous slide. In our case, we identified dozens of enzymes with superior activity, and we're also able to get our best enzymes from the screen back into our customers' hands very quickly. Had we stuck with smaller batch sizes emblematic of cloning by hand, we would have had to go through many more cycles of discovery or engineering, and arguably we never would have gotten to the enzymes that we identified in this screen. Furthermore, these screens provide an enormous amount of data that we can use for refining our design tools down the road. I've mentioned protein engineering several times in the context of enzyme discovery. On the previous slides, I was showing primarily enzymes sourced from metagenomic sequence databases. Often, we'll follow up this enzyme discovery work with additional rounds of protein engineering to further improve activity or expression of our lead enzymes. We've also begun using synthesis to do both protein engineering and enzyme discovery simultaneously. That is, we can modify our starting enzymes in silico to introduce mutations that are suspected to positively impact activity. Because we're making these libraries via synthesis, we can simply encode all of these changes digitally and then order them. Twist also has a variant library service for ordering libraries of mutated enzymes. This allows us to design smarter protein engineering libraries that leave out variants that we know will be inactive or mutations in regions that we want to conserve. As we've collected data on the performance of tens of thousands of enzyme sequences over the past few years, we've developed machine and deep learning models based on these data. In fact, the way that we map enzymes is now driven by deep learning. So in these figures, enzyme networks are clustered based on a neural network designed at Ginkgo. As I mentioned at the beginning of the talk, Moore's Law empowered the software industry to pursue ambitious projects on the basis of exponentially improving computer hardware. In biology, we've seen a similar phenomenon happening with sequencing, where more ambitious sequencing projects are being pursued every year. Hopefully, I've given you a sense of how we're doing the same with DNA synthesis at Ginkgo. Because we expect the scale of DNA that we can design and order each year to continue to increase, we can plan on when and how we'll gather enough data to enable more advanced applications of deep learning. From my perspective, we're only just beginning to unlock the potential of the genomics revolution that started around the year 2000. Essential to unlocking all the sequencing data that now sits in sequence databases is our improving ability to computationally model and, importantly, synthesize sequences of interest. This has to be done at large scale in order to collect enough data to train machine and deep learning models. And while I've primarily talked about enzymes today, I think this holds true in all facets of biology, research, and development. And just to zoom out a bit from enzymes, we also work on engineering and optimization of metabolic pathways, and of course we also work on making genome-wide changes to the microbes that we engineer as well. Synthesis, again, factors heavily into these strategies. For example, we can make fewer compromises in construct design based on avoidance of certain restriction enzyme sites, for example, needed for assembly. Synthesis allows us to make genes without scar sites when that's essential to strong expression. Furthermore, the scale afforded by synthesis, as well as the automation applied to all aspects of bioengineering at Ginkgo, means that we can take more shots on goal in each round of engineering. In practice, we found that it allows our engineers to try design strategies that may have been too risky to attempt if they were only making a small handful of designs and working iteratively. This has paid off in pathway design, where we've been able to simplify how we design bacterial operons. To wrap up, hopefully I've illustrated how large-scale DNA synthesis dramatically changes how biology is engineered. With respect to sequence design, it's very convenient to work with digital sequences. With synthesis, you can make exactly the sequence that you want to test in most cases. Restriction sites, PCR troubleshooting, and sequencing analysis can all be avoided by ordering sequence-perfect clones. We've also found that we can use sequencing data to identify enzymes from sources that would be impossible to clone from and make them via synthesis instead. From a protein engineering perspective, variant libraries offer a lot of advantages. You can specify exactly which positions you want to modify and which amino acid substitutions should be allowed at those positions. This allows for the design of smaller, smarter libraries for screening. Since Ginkgo engineers everything from bacteria to mammalian cells, we have a lot of different vectors that we need to support. Twist has onboarded these vectors so we can use synthesis for a wide variety of organisms. As long as we can transform an organism in high-throughput format, we know that we can deploy a lot of content from Twist into those organisms. If you have a challenging organism or a specialized assembly strategy, non-clonal Twist fragments are also a fast and cost-effective input to DNA assembly. And most importantly, I hope I've convinced you that trading valuable FTE time spent cloning for larger scale experiments helps collect better data in less time. I mentioned that we've used synthesis to find new enzymes from unclonable DNA, and I think this is a fun example of how synthesis can be used. So this photo is from last month's Scientific American, which features an article about a project we did to recreate the fragrances of extinct flowers. So for this project, we worked with the Harvard Herbarium to sequence DNA from plant material that had gone extinct. As you might expect, this DNA was highly fragmented, and it would have been impossible to clone intact genes from this material. Also, since these plants represent the last remnants of extinct species, we could only afford to take very small samples for sequencing anyway. Instead, we were able to computationally reconstruct the fragmented genes from our sequencing data and then synthesize the library of those reconstructed enzymes. Ultimately, these enzymes produced fragrance molecules that were used by perfumers to create a concept fragrance based on extinct flowers. Anyway, there's a lot more about this project in the Scientific American article, and I encourage all of you to check it out there. And on that note, I'll stop here and I'll open up for questions. Thank you.
But before we get to Patrick's presentation, I want to encourage everyone to submit questions for our Q&A session that will begin right after his talk. We'll try to answer as many questions as we can, so just click the Ask a Question tab on the right-hand side of the presentation screen, type in your questions, and hit Submit. All right, with all that said, let's begin our webinar and hear what Patrick has to say. Thanks, Patrick. That was a beautifully laid out presentation with some excellent data highlighting some exciting projects at Ginkgo Bioworks. Before we start the Q&A session, I want to let everyone know that this is one of their final chances to submit questions for our speaker. So hurry up and send them in now. Since it looks like we have some really great questions that have come in already, let's start the Q&A session and try to get to as many questions as we can. Bear with us for just a moment and we'll begin the Q&A.
Hi everyone, this is Kevin Davis, the editor-at-large for Genetic Engineering and Biotechnology News, stepping in for Jeff to handle the Q&A segment of our webinar. I'm also the author of The $1,000 Genome, so it's been great listening to Patrick talk about the remarkable advances in DNA synthesis and genome synthesis in the wake of the era of next-gen sequencing. So we've had a flurry of questions, and we're going to dive in and try to get in as many as we can over the next 15 minutes or so. And we'll start with Zach from Bluebird Bio, who asks Patrick, how far off do you think we are from genome synthesis? I guess he's referring to mammalian genome synthesis.
Yeah, that's a really interesting question. I think in terms of bacterial genomes, if you look at the volume that Twist is shipping, and certainly the volume that we're ordering, we're receiving more than a bacterial genome worth of DNA every month now. So I think in terms of genome synthesis is really a question of how complex is your genome and what are you trying to accomplish by synthesizing a genome de novo. I would point to the Yeast 2.0 project, which is a global collaboration to build full yeast genomes. That's something that's pretty cutting edge, but could become much more accessible as the technology improves. So I'd say we're there today for microbial genomes. We don't really do a genome synthesis for our projects at Ginkgo. We're more in the process of rewriting portions of genomes, but it's certainly something that's technically possible today for microbial. And mammalian, there is a similar project for the human genome that's trying to get underway over the last year or so. So I'd say it's 5 years off.
Yep, the HGP Right project. Yeah, exactly. Next question. What was the impact on the team's efficiency after connecting your systems through the API that you referenced? Do you think companies in our field undervalue the importance of it when working with high throughput?
Yeah, that's a great question. I think for us, we really value the time of our scientists and engineers. And our volume was getting to the point where, the original way that we would send sequences to Twist was to bundle things up as CSV files and email them over to the Twist team. Our volumes are getting such that we're actually going over the attachment limit for emails. And as you can imagine, there are a lot of other features that the API adds that save us time in terms of doing complexity checking and checking in on orders. So it replaced both a lot of hands-on time in terms of sending over orders at the volume that we're working, and also saves we had a call every week just to stay up to date with orders, and that the need for that call was significantly reduced just by having the automated checks we were able to perform through the API. So yeah, it's really about a time savings, and it was many hours per week saved.
Okay, great. Michael from Harvard, what if you can neither synthesize nor recode a DNA sequence, for example, promoter sequences?
Yeah, so there are certainly limitations to DNA synthesis. If you think about it, what biology is doing when replicating DNA is that it's doing template-based copying. So certain regions of DNA are hard to synthesize de novo, either due to GC content or repetitiveness. We've certainly run into that here as well, although I'd say that with the complexity checker that we use through the API, we're usually able to query sequences and redesign them for synthesis if needed. What we've done when we have promoter sequences or other sequences that are essential to a design, what we'll do is we'll clone those into the vector backbone itself and then set up custom vector with Twist. And that way you can preserve regions of high complexity or high GC content or low GC content for that matter, while still being able to synthesize the majority of what you need. In practice, we've also been able to down select on parts that we use based on things that are friendly with synthesis, and that has made our jobs a lot easier, because then it takes a lot of the guesswork out in terms of needing to figure out if a certain part is compatible with synthesis or not.
Okay, Patrick, thank you. Next question. In our lab, a mammalian synthetic biology lab, a project generally requires making 500 different plasmids and takes about 3 years. Would this scale of throughput benefit from DNA synthesis?
Yeah, so 500 plasmids would be a small set of designs for us. So as I mentioned in the talk, each shipment we get from Twist comes in a minimum of 384 well plates. And the other advantage to synthesis is that you don't have to do things serially. You can essentially order an entire plate for 500 plasmids. You could get that in a single order and have those all ready to go in about 3 to 5 weeks. So I think that's the time savings of approaching that in parallel, added with the labor savings of not having to do all that cloning and serially. Just frees up a lot of your research time to do other things.
Sounds like they should talk to Twist. Next question, what resources do you recommend for a student trying to build high-throughput biofoundries?
Well, so in terms of answering that question, it really depends on what you're building that foundry for. In my talk, I mentioned that there's quite a bit of capabilities that we've built around high throughput synthesis to support the work that we're doing, which is engineering organisms. So I would say it really depends on what fits into your design build test cycle. I would say if you're really interested in pursuing this, studying and writing code is essential. Being able to specify designs via an API, we do it in Python, for example, helps you think at the scale that you need to really run into the issues you may discover trying to set up high throughput pipelines. So I'd say focus on the software and also the hardware that you may need to put together to achieve the pipeline that you have in mind.
Okay, quick reminder to the audience, please get in your questions before the top of the hour. We'll try to get to them, and we'll prioritize them if they're really good. Next question for Patrick is a little ambiguous, Patrick, but you may be able to see what the question's intent is. How far do you think we are from code to proteins, including transcription?
Yeah, well, so the the analogy we like to use is that biology is fundamentally runs on digital code, which while I think we can show is true, right? If you change a DNA sequence, you get a different biological output. The challenging part of that is that it was code written by evolution, so we don't have nice commented code to understand how all that was put together, right? I think in terms of the way that we've approached engineering biology, one of the reasons that we're interested in doing such high throughput work is not only to accelerate the progress that we can deliver for our customers, but it's also so that we can learn a lot about what design strategies work and what design strategies don't work. I'm afraid I don't have an answer for you, because being able to specify exactly what you want an organism to do depends on the complexity of the task and what you need. I would characterize what we do at Ginkgo today as we've built a highly automated rapid prototyping facility that lets us sift through thousands of designs quickly. The future is to be able to soon go through hundreds of designs and maybe tens of designs. But we're just not there yet, which is why we're so focused on high throughput screening, so we can collect enough data to do low throughput experiments in the future.
Okay, very good. Next, how do you maximize the likelihood of success when choosing pathway variants?
Yeah, so for pathway variants, a couple of things. One is de-risking your approach. So again, having a large number of shots on goal certainly helps. When selecting a pathway architecture, we have sets of designs that have worked well in the past for us over the years. So we have sets of designs that we will always default to, but we'll always also make sure to cast a wide net in terms of design. So for example, we've learned a lot in the last few years in terms of how to synthesize and optimize operon architecture, for example, and we'll preferentially group genes into operons based on designs that we've developed. But that's really something where it's still not one shot on goal. We try to prioritize designs that work well in the past, but make sure that we're screening enough designs that we can discover even better designs that we may not have thought about initially.
Okay, two questions, I'll take them together, asking about error rates. Error rates per construct that you receive from Twist. What do you observe, and how many clones do you have to sequence to get the sequence verified? Let's take those together.
Yeah, so most of the content that we get from Twist, and definitely the content that was presented in this webinar, is received as sequence perfect clonal DNA. So we receive that DNA and move it straight into screening without sequence verifying it. Because things are moving through a large number of robots and there are many liquid handling steps involved, we will often spot check via sequencing at the end of our screen to verify that there were no well swaps or anything like that. But effectively we just trust what we get from Twist, and that's been borne out in our spot sequencing over the last couple of years.
Okay, great. Good question coming up. By automating some of the typical lab work done by hand and ordering genetic code in bulk from Twist, it sounds like you've removed two big bottlenecks from genetic engineering. What do you think the next bottleneck will be?
Yeah, well, so while we work towards the future of not having to screen, I think I'm always going to come back to the need for better and faster screening methods. I didn't have time to discuss this in detail today, but we've invested a lot in a variety of technologies that give us systems biology detail about organisms. So all the various omics, from transcriptomics to metabolomics to proteomics. And we're always hungry for more data. So there are a few ways around that. One, which is faster instruments, and then the other, which is genetic selections or multiplexing technologies that allow you to increase your throughput for instrument. And short answer is that we're pursuing all of those approaches.
Okay, thanks. Sean says, greetings, great presentation. Thanks. We have an AI question. Thank you. Using your AI tool, is it possible to predict new enzymes and activities that currently do not exist in nature?
Yeah, that's a great question. I think enzymes are exquisite catalysts, and there are many examples of leveraging enzymes to catalyze reactions that may not necessarily be found in nature. For approaches like that, we will take quite similar discovery approaches where we will try to seed our search with enzymes that we think have related activity. And oftentimes we're able to identify enzymes with that activity on novel substrates. So again, it's part numbers game, but it's also a prediction game. So one of the things we like to do to train our machine learning tools is to look at many different substrates for an enzyme family. And that can help us differentiate specificity in terms of substrate size, for example. So I'm afraid I can't give much more detail than that, but certainly anyone who's worked with the machine learning knows that these approaches are very hungry for data. So we try to parameterize our models with data on multiple substrates and many different enzyme types.
Okay, Patrick, we're close to the end of our webinar, so we'll enter the rapid fire round here with a few final questions, and I'll just ask you for some pretty succinct responses. Ginkgo's success showcases the importance of a Foundry and high-throughput automation into SynBio workflows. Are there unique characteristics of Twist DNA that makes it really well suited to automation?
I would say scale and being able to deliver in batches. Through the years at Ginkgo, we've evaluated a large number of providers, and I'd say what we really like about Twist is that when we order thousands of things, they arrive within the time window we expected and show up in 384 roll plates with the barcodes we expect. So being able to deliver in large batches rather than one tube at a time is certainly something that differentiated them.
Okay, thank you. When engineering a pathway or molecule, what is the typical number of variants that you aim for in every step?
Yeah, so that's a great question. I think really what we try to do is if screening is in fact the rate limiting step, then we maximize the use of that screen. So it can be anywhere between a few hundred variants to tens of thousands of variants depending on throughput. We try to make the size of the library dictated by what can be screened per batch rather than the amount of DNA that we want.
How do you decide when you're done improving the molecule you're working on? Or are you never done?
Yeah, so well, so it really depends on the project, but we work with our customers to set technical benchmarks that we want to hit. And as you said, you may never be done in terms of you may hit those benchmarks to go to commercial scale, but may want to continue to do improvements to extract more value from the organism. But that's something that we work on on a case-by-case basis with our customers.
It's an interesting question. What did you think was important in target selection that perhaps turned out not to be impactful on your success rates?
Well, starting out, I thought we could trust automatic annotations of genes in genome databases a lot more than we do now. Fundamentally, what we've seen is that gene annotation is overfit to a small number of genes that are characterized. So we need to develop our own annotations in-house.
All right, I'm going to close with two final questions. Patrick, thanks so much for doing this. To what extent do you use and reuse libraries of standardized regulatory elements such as promoters and previously an RBS in genetic designs at Ginkgo, or are they designed ad hoc by your automated genetic design algorithms?
So the answer is both. So certainly for many of the organisms that we work in, we don't need to reinvent the wheel with parts, but we do also regularly add new parts and designs as well. But certainly very useful to have reusable parts, even if you're not assembling them de novo every time.
And a nice, appropriately big picture question to close. How far away are we from de novo building enzymes and proteins that are completely synthetic?
Yeah, I mean, certainly there are many efforts in the de novo protein design world to point to. I would actually turn the question around and say, you know, what is the limit to what we can build with the building blocks that evolution has already selected for? So certainly, like, despite the fact that we're not discovering a lot of new protein folds, if you look at the protein folding, community, we are discovering lots and lots of new novel arrangements of those folds that allow lots of activity. So, even if the enzymes that we end up developing look quite different than natural enzymes, we're still learning and borrowing heavily from what evolution has brought us. So I'd actually turn around and say, like, how de novo do we actually need to get to be able to synthesize arbitrary molecules with enzymes? And I think I think the next 5 to 10 years are going to be very interesting, but we've certainly been impressed by what enzymes can do already.
Great. Well, I think we should leave it there. I know we didn't get close to addressing every question that came in from the audience, and I apologize for that. But I'm sure for those folks who did ask a question, you can take it offline, either to Patrick at Ginkgo or Patrick Boyle, or to a contact at Twist. I'm sure they'd be more than happy to try to take some of those more technical questions offline and help you out. So with that, we've come to the end of a really engaging webinar. It'll be archived on our website at Genetic Engineering and Biotechnology News, so you can watch it on demand for at least the next 12 months, or forward the link to friends and colleagues who were unable to join today. So all that remains is for me to thank our guest speaker, Patrick Boyle from Ginkgo, who did a great job, Twist Bioscience for sponsoring this webinar. We really thank them. And most of all, however, we thank you, the audience, for joining us and staying for the full course of the webinar. On behalf of the entire team at Genetic Engineering and Biotechnology News, I'm Kevin Davis. We hope to see you on another Gen webinar in the very near future. So thank you and goodbye for now.